Hello friends, in today's video I'll tell you how to easily learn glycolysis or Embden-Meyerhof-Parnas pathway or EMP pathway. So let's start. The first compound is glucose. Then it is glucose 6-phosphate. Then it comes fructose 6-phosphate. Fructose 6-phosphate converts into fructose 1,6-biphosphate. Then it undergoes splitting. The splitting leads to DHAP, that is dihydroxyacetone phosphate, and PGAL, that is phosphoglyceraldehyde. Now, phosphoglyceraldehyde converts into 1,3-bi-PGA, that is 1,3-biphosphoglycerate. 1,3-biphosphoglycerate gives 3-phosphoglycerate. 3-phosphoglycerate gives 2-phosphoglycerate, which again gives phosphoenol pyruvate. Phosphoenol pyruvate converts into pyruvate or pyruvic acid. Here it undergoes 10 steps. You will mark it into 10 steps. First, second, third, fourth, fifth, sixth, seventh, 8th, 9th and 10th. Then it becomes easy. Now we'll learn the enzyme. The first enzyme is hexokinase. So we have written the first enzyme as hexokinase. It can also be called as glucokinase. Now we'll write the next enzyme, that is phosphohexoisomerase. So let's write it. Now from fructose 6-phosphate to fructose 1,6-bisphosphate, we'll write the third enzyme, that is phosphofructokinase. Here it is there fructose, so we'll learn it as phosphofructokinase. Now here it undergoes the splitting. So for splitting, the enzyme is aldolase. Now for DHAP to PGL, the enzyme is triose isomerase. Now from 3-phosphoglyceraldehyde to 1,3-bi-PGA, the enzyme is glyceraldehyde phosphate dehydrogenase. Now from 1,3-bi-PGA to 3-PGA, we'll get the enzyme phosphofructokinase. From 3-PGA to 2-PGA, the enzyme is phosphoglyceromutase. Now from 2-PGA to PAP, we have enolase. From PEV to pyruvate or pyruvic acid, we have the enzyme pyruvate kinase. To learn the last 4 enzymes, we can learn it as kinase, mutase, lase and kinase. It will be easy for you to remember. Now it comes the learning of the ions like magnesium and which steps are reversible, where it converts from ATP to ADP or ADP to ATP, and where it undergoes the removal of water. So first we will see which steps are reversible. So step 2 is reversible and step 6 is reversible, so we'll mark it as reversible. Step 6. Now step 7 is also reversible, 8 is also reversible, and the 9th step is also reversible. Now in the 6th step, NAD converts to NADH plus H plus, so we'll mark it. Now we'll see where we get magnesium. So in the first step we get magnesium. In the second step also we get magnesium. In the third step too we get magnesium. In the seventh step, in the ninth step, and so in the last step also we get magnesium. Now we will see where ATP converts to ADP. So in the first step ATP converts to ADP, and also in the third step ATP converts to ADP. Now we will see where ADP converts to ATP. So this happens in the seventh step. And also in the 10th step. Now in the 8th step the removal of water occurs. In the 6th step, along with NADH plus H plus, we also get a phosphate. So here we'll mark it as phosphate. And also in the 10th step, along with magnesium, we get potassium ion. So with this, we have completed our glycolysis pathway or Emden-Meyerhof-Parnas pathway. If you write the numbers where we get the ions or where we get ATP from ADP, it will be easy for you to learn. Let me show you. So we have seen that in the step 1, 2, 3, 7, 9 and 10 we get magnesium ion. In the 10th step we get potassium ion. ATP converts to ADP in the first and the third step. In the step 2, 6, 7, 8, 9, it is reversible. 6th step, NAD plus converts to NADH plus H plus. Also we get phosphate. In the 7th and the 10th step, ADP converts to ATP. 8th step, removal of water occurs. With this, we complete the glycolysis or the Emden-Meyerhof-Parnas pathway. So, if you prepare an index this way, it becomes easy for you to learn. First, you'll write the compounds, then you will write the enzymes, and then if you learn the ions and ADP where it converts to ATP, then step by step it will become easy for you to learn. Did you like my video? If you like my video, please share
and do not forget to subscribe our channel.